Um, I'm going to begin today, so we're relatively close to starting time. We do have people who join us by webinar at 1 o'clock, so it's important for us to start. I want to welcome everyone in the room and welcome those that are online. We are going to be giving a presentation. 4 of the committee members have sort of parsed out the findings so you can hear what we have found in a year and a half of intensive debate and research. But if you have questions that emerge as we present, you should hold your questions and then at the end go to the microphone to ask the questions. For those who are online, there is a website or an email site, builtmicrobiome@nas.edu, that you should be emailing your questions to, and then one of the staffers will go to the microphone and ask the questions for you. So all of this is being recorded and will be available for those who are not able to attend today. So. And for sharing. Let me start by giving you just a quick insight into the committee. I had a privilege of being one of the building science members of the committee, but the majority of the committee were public health and medical researchers and medical practitioners who are dealing on a day-to-day basis with the health implications of microbiomes, which we'll talk more about. And the challenge to us, which I'll give a little more insight into, was to really uncover the links or the research needs that would uncover the links between the built environment and the microbiome. Yeah, this is giving you the list. The 4 of us at the podium are in red, so you can see who will be speaking today. So one of the first questions is why understand the nexus of humans, built environment, and microorganisms? And we spent the reason this is so important is because we spend a majority of our time indoors. It's even more than 90%. And buildings house a wide variety of microorganisms that form dynamic microbial communities. And that's actually an issue, which is you can't tease this out piece by piece. These are actually communities of microbes. Some of them are good microbes, which we spent a lot of time talking about as good microbes and harmful microbes. The built environment microorganisms include bacteria, and with that, bacterial infection, viruses, viral infections, and fungi, which things like mold that lead to other respiratory and other kinds of infections. They could be living microorganisms. They can also be dead and still have an impact, or they could be dormant and be brought back to life. Sounds like some late-night TV show. The microbial research that exists does span quite a diverse set of disciplines, and we actually spend a lot of time talking about the need to bring those disciplines together, especially when you cross the boundaries between the built environment and health and humans and how humans behave. And we also found that there is a mixed set of tools out there that are emerging, and old tools and new tools, and a need for common repositories and common research methods. And we'll talk about each of these in terms of our recommendations in the next half hour. Just to give you the most simplistic sort of the triad of issues, the blue bubble here is the most important one, which is to understand what are microbial communities, and again, bacteria, viruses, fungi, and other lower eukaryotes, and the mix that can occur between them. And understanding the built environment that may be supporting those, both good and bad microbes. And we spent time talking about the building enclosure from roofing and siding, especially in strong storm events like right now. We know that fungi, as a result, is going to be a major issue in storm areas. We talked about the mechanical systems and what they do to both viral and other dissemination. We talked about plumbing systems and the way in which we bring water into buildings, and then interior systems, and of course how humans operate buildings and what they do in the buildings. So the built environment, we spent time going through what are the decisions in the built environment that really might be the most critical to a healthy indoor microbiome or to a harmful one. We broke our study in the built environment into 3 categories: things that are airborne, things that are waterborne, and things that are human-borne and then exchanged on surfaces. And of course, humans are involved in all 3 of those, but ultimately surface exchange is typically from human to human. The outcome of human health, we spent most of our time talking about known medical conditions, but we also spent time looking at the psychological or neurological, behavioral, some of the other variables in the human occupants that might be critical in the long-term study of microbiomes. So just a simple statement of tasks that was given to us by the sponsors was to examine the formation and function of microbial communities in the built environment specifically, to assess what's known about the complex interrelationship among microbial communities, humans, and the built environments, to explore implications of this knowledge for building design and operations. So we're really trying to get information back to the building community to design and build and operate buildings more effectively, to articulate opportunities and challenges for practical application of improved understanding of the indoor microbiome, to identify a set of knowledge gaps and prioritized research goals for the research community, and to recommend additional actions to advance an understanding of microbes in the built environment. With that, I'm going to pass this to Diane Gold.
Thank you, Vivian. So one of the tasks of our committee was to review what we knew and what we needed to learn about how indoor microbes affect us. We noted that there is a demonstrated link between exposures to infectious microorganisms in built environments and human health, that there's evidence of a link between exposure to indoor microbes and either the worsening or development of respiratory and allergic symptoms, particularly those arising from exposure to microorganisms that flourish in damp indoor settings. There's preliminary evidence that suggests certain microbial exposures may have beneficial health effects, such as protection from allergy and respiratory symptoms, including early life exposures to diverse microbes associated with animals. However, impacts on non-respiratory outcomes, such as neurologic impacts, are less well understood. And many open questions remain. They include, what mechanisms underlie health effects? By mechanisms, we mean the way individual or communities of environmental microbes, be they alive or fragmented and non-viable, cause positive or negative effects in humans. We ask, how do factors such as stage of life, route, dose, genetics, or health status affect responses or modify responses? For example, when we ask about stage of life, we ask, do infants without established allergy or asthma respond differently from adults with established allergy or asthma to the same microbes or microbial components? When we ask about route of exposure, for example, we would ask, if environmental microbes are ingested by toddlers as they crawl on the floor, do they have the same effects as if the toddlers were inhaling the microbes? Does the upper or lower airway have different immune responses to environmental microbes than the gut? And in asking about how genetics may affect or modify responses, we ask, can genes, perhaps in combination with other environmental cofactors, modify how environmental microbes affect people? Learning about mechanisms of action of microbes, about who responds and how, may inform building to improve health for everyone in the built environment, as well as development of targeted protective strategies. Another open question is, can the building microbiome impact the human microbiome, and what does that mean? That is to say, can the building microbiome influence what organisms grow on the skin, in the nose, in the mouth, in the airways, or in the gut? And finally, another open question is, what about other health effects other than respiratory or allergic? As Vivian has mentioned, neurocognitive health effects are an area that is less well understood, and more work is needed to understand the contribution of indoor microbes to neurocognitive outcomes. And so with this, I'm going to turn you over to Jordan.
Thank you, Diane. Two of the chapters in the report dealt with what we know about microbes in buildings. Thank you. What we know about two of the chapters in the report dealt with what we know about microbes in the buildings, and also with what we know about how we can encourage certain microbes to be in our buildings or discourage certain microbes from being in our buildings. So there's a cycle of life for a microbe in a building. They can enter via cracks in the building envelope. They can enter through ventilation. They can enter on people. Once in buildings, they can enter on dogs. Once in buildings, they can grow if there's enough moisture. And when they grow, they can produce metabolic products that can be emitted then into the building. And microbes also die in buildings. They can be emitted through ventilation systems, but they can also die leaving pieces of microbes around as well. So there are physical attributes and environmental attributes and occupancy attributes in buildings that dictate what the concentration of microbes in buildings are and what the ecology of microbes in buildings are. So, for example, microbes that are found throughout indoor air, they're found in buildings, plumbing systems, they're found in standing water sources and on building surfaces. And it's things like outdoor environment and it's and the climate and building site selection, which in part dictates what are the microbes inside of the building. There are other building factors including airflow, temperature, humidity, chemicals, and the maintenance and cleaning of the building. And then occupants always loom large in this equation. Occupants like animals, pets, humans, pests, and even plants can dictate the content and the ecology of microbes in buildings. So there are a few other factors. Some of them help microbes survive in buildings. For example, indoor humidity can influence the survival of specific types of pathogens and non-pathogenic organisms in buildings. Surface moisture characteristics like water activity can dictate at points at which microbes may grow and thrive in buildings or at which microbes can't grow and thrive in buildings. And then surfaces, it's important to point out, serve as strong reservoirs for microbes in buildings. So probably some of the microbes that you're inhaling right now came from some surface in the building that was disturbed when we all walked in today and was aerosolized. Microbes can also affect the building themselves. For example, if you know of a rot in a building that's caused by moisture, microbes are always in that equation, increasing the deterioration of building materials. Another example would be biofilms on the cooling coils of an air conditioning unit. The biofilms reduce the heat transfer efficiency. They also can choke off some airflow and overall reduce the efficiency of those types of systems and increase energy use. Well, whether we like it or not, we are designing buildings and we are occupying buildings and we are maintaining buildings in such a way that is affecting the microbial community. But we do know some things about how we might maintain and occupy buildings and treat buildings so that we can dictate maybe some of the microbes that we want in the buildings, both encouraging good microbes and discouraging the bad ones. And so in another chapter in the report, we looked at these types of interventions. There are physical interventions like changing in ventilation rate and filtration. There are also chemical types of interventions such as producing antimicrobial surfaces or the use of cleaning products. And then a third part of this table says environmental probiotics. And we're still thinking about the physical and chemical interventions here, but it's a recognition that these interventions need not only be to control bad microorganisms, there can be a situation where we want to control or we want to encourage the presence of specific types of microorganisms in the building to improve our health. And it's still going to be some of these similar interventions that are capable of doing that. So thinking about some tools to evaluate these interventions, we need quantitative frameworks and better quantitative frameworks to evaluate how we can design buildings and intervene in such a way to encourage healthy microbial exposure. One thing that we've talked about a lot in the report is that these interventions and these models shouldn't act on their own, but we should account for the trade-offs that occur when we make such an intervention. And that means to systematically not just be thinking about human exposure to microbes, but to be thinking about the energy and the economic costs that these buildings interventions may pose. There are lots of knowledge gaps in this arena right now. So for some examples, if we think about mold in buildings with excessive moisture, the way to perhaps treat that would be to remediate those buildings, but what level do you remediate it to? You're supposed to remediate it to something called a normal microbial ecology, which sounds like a good idea if we could just figure out what that normal microbial ecology is. We need to understand what constitutes a good indoor microbiome. It's going to be tough for us to implement any building interventions until we have a much better understanding of the microorganisms that we want to have around us for healthy occupancy. We need to test our health-relevant hypotheses and understand more whether there's biological mechanisms between some of the things that we see, like, for example, higher air exchange rate or ventilation and better health. And we need to enhance foundational areas to support this research structure, better ways of sharing data, better analytical and modeling approaches as well. More fundamental research to understand things like microbial emission rates and particle size distributions. And then finally, in developing this infrastructure and practices, we need to work much more with those that are actually designing and doing the building of buildings. So if we think about a vision for the future in this area, we think about transmitting it from, or transforming from, a situation where we have now where we occupy, design, and produce buildings, and we affect the microbial ecology, but in unintentional and unknown ways, to buildings where we can apply our knowledge about microbes, built environments, and humans to inform the design, operation, and maintenance of buildings. I'll hand things off to Jonathan next.
So the next part of this presentation is articulating how we achieve this vision. And so basically the report decided to try and summarize a set of research objectives that can be pursued to achieve and answer these questions. So the question posed here is, how do we get to this future vision in which we understand enough about the microbial communities to manage these indoor environments? So this research agenda that's articulated in the report is organized around these 5 major objectives, and that are then broken into 12 priority areas to accomplish those 5 research objectives. And here I'll just sort of articulate them sort of at a high level and then go through each of these areas in a little bit more detail. But moving sort of counterclockwise or clockwise here, so the 5 major objectives are characterizing the interrelationship, so not just looking at microbial communities or the built environment or human health as separate entities, but looking at the relationships and how those 3 different entities influence each other. The second sort of major area here is really kind of categorizing healthy microbial communities from those that are unhealthy and how that affects human health outcomes. The other objective here is to recognize that there's more than just the human health impacts, and that's sort of the building maintenance, energy requirements, and other impacts affected by the microbial communities. And then also there's a major challenge in terms of, as we develop this knowledge, how does it actually get translated into practice in terms of managing building maintenance and design of new buildings. And finally here, there's sort of this idea of that we really need to do, improve our ability to manage the research infrastructure, given the complexity and the interdisciplinary nature of this research, that more infrastructure is needed to support this research. So stepping through these in a little bit more detail, so each of these categories or 5 broad objectives are then mapped to these 12 specific recommendations. So to characterize the interrelationships among the microbial communities and built environments for systems of air, water, surfaces, and occupants, there's sort of these 2 major sort of specific objectives that are presented. So improving the understanding of the relationships among the buildings with microbial communities. So this is, how does the building affect microbial evolution and ecology, so that we can ultimately start to build models of how these microbial communities evolve in different conditions. Another major factor that's been understudied to this point is better incorporation of social and behavioral sciences to analyze how people impact the environment in these built buildings that we are occupying ourselves. The next area here is sort of assessing the influence of the built environment and indoor microbial exposures on the composition and function of the human microbiome, and on human functional responses and on human health outcomes. And this is sort of organized into 3 objectives or 3 research suggestions. The first of which is really around this idea of that more emphasis should be placed on developing different types of study designs that can support each other. So the idea that there's complementary study designs are needed. So in addition to the longitudinal observational studies that can be taken over time to sort of observe how microbial communities are associated with different conditions of different building environments, as we sort of identify specific microbial community compositions that are of interest, we need other more intentional study designs that can then see how those microbial community compositions are impacted in terms of their exposures to humans and how that affects our health. So for example, more precise animal studies may be needed to actually study the route of entry and exposure for a particular set of organisms and how that affects infection of the human ultimately. Another key component of this is to better disentangle sort of how different stages of human life are impacted by these microbial communities. So early stage exposure versus late stage exposure in life, as well as genetic susceptibility to particular microbial exposures, so the host genetics. And finally, there was a recognition that not just the microbial communities themselves are important to study, but we have to also understand the chemical exposures and their relationship to the microbial community, so that it's co-infection that can potentially have an impact on functional responses. So the next category is the idea that was previously mentioned on essentially the non-health impacts of interventions. And this is in this area of understanding, as we try to modulate the microbial communities of built environments, there are other consequences that we have to be aware of. For example, if you increase airflow, you may require greater energy consumption, and also as you increase moisture to support community growth, you may have building degradation or material degradation introduced. And so those kinds of consequences have to be investigated and understood. So in the category of advancing research infrastructure for addressing the microbiome built environment questions, there's these 5 different categories that are identified. So number 7 here is refining tools and methods for elucidating microbial community identity, abundance, activities, and functions. And this has been an area of major development, particularly in the area of genomics and understanding the taxonomic composition of organisms in the community. But there's a recognition that more needs to be done in terms of better quantifying absolute abundance of microbial constituents in a community, and particularly functional activity of these organisms. Number 8 here is refining building and microbiome sensing and monitoring tools. So there's needs to be continued effort at developing sensors in terms of humidity measurements at surfaces and in appropriate areas for collecting the parameters needed to characterize the buildings and the environment. In addition, developing guidance on sampling, so where to sample and what are the most relevant metrics to capture that will be relevant to exposure to humans from the built environment. Number 10 here is developing a data commons. So one of the recognitions is that there's a tremendous amount of complex data here that is, there's many groups that are generating measurements, particularly molecular biology measurements, genomics, transcriptomics, and there's a need to actually be able to have a common set of tools and storage areas where we can actually deposit this information, this data, and have it more widely accessible so that we can endeavor to build more understanding across multiple studies and start to share sort of research findings and connect to other studies from multiple building environment or built environment studies. And finally here is this topic of developing new empirical, computational, and mechanistic modeling tools. So we need to be able to better link this data collection efforts to actually building predictive models to help predict outcomes. And so finally, the last category here is translating research into practice. So ultimately, I think as the knowledge is being developed here, there was a clear recognition that there has to be major efforts to understand how you transfer that knowledge into actual practice to the practitioner to actually implement this new development. So yeah, this can take on the form of developing new guidance for professional building designs and operation, new guidelines, and potentially guidance for clinical practitioners and information for building occupants, and actually motivating individuals, the residents of the buildings, to actually take this information and make some use out of it. And with that, I'll pass it back to Vivian.
So I get to do the wrap up, and then we can open up for Q&A. The key issues for us have been not just to put together a set of research recommendations, but to really try to understand the power of a collaborative sort of a national commitment to understanding microbial environments, both good and bad, to increase the access to data and knowledge, to facilitate comparisons, to, as such, inform the design community and the building management and operations community into what are microbial communities and what actions they take that can have the biggest impact. And just the process of this committee, which I go back to give you the list of this committee members, we met over a significant length of time, both face-to-face in a number of venues. We had 4 public information gathering sessions in DC and California. We invited 45 experts to present their insights so that we had not just the power of the committee, which was an incredible group of researchers and practitioners, but 45 others who've been active in this space of microbes in the built environment. We also had a host of teleconferences to try to iron out and debate what were the research recommendations, what were the gaps in knowledge, what were the linkages that were known, and what were the research recommendations. All of the presentations that were made are publicly available, so you can see those 45 presentations, and the videos are on the study website, where you can also download a copy of the study itself. I just want to stop for a minute here just to talk about some of the insights that, as we began to debate this, that really were exciting to me and important to me as a practicing architect and someone who's actively involved in the performance of buildings over time. was just to begin to understand how design decision-making and building operation decision-making can make a huge difference. And examples that I think would be meaningful to the people in the audience would be things like Legionella, which everyone has heard of and knows it as sort of a scary phenomena. But just the fact that our plumbing systems and our cooling towers, of which we merrily put more and more cooling towers across the country, are natural breeding grounds for Legionella, and the importance of really studying and maintaining those facilities, because we need, if we need air conditioning, which is, of course, one of the big debates, is can we begin to move away from cooling towers as the most standard or solution set. We also spent quite a bit of time talking about air and the air systems in buildings and the inadequacy in many cases of ventilation systems. We talked about hidden spaces and the fact that in buildings we encapsulate a lot of things. Our ducts are hidden spaces. Once they're put in place, we have no clue what's inside those ducts, and the chances that we get back into them are very small. And hidden spaces inside the walls of our buildings that. So there's a whole set of issues related to the airstreams that we have set up and the inability to actually sort of monitor or even maintain those airstreams. And then even in the ones that we can see, in the surfaces that we can see, we don't have a standard set of practices for what happens when we see mold or mildew appearing on walls. Should we have hands-free entries to classrooms so that kids are not all shaking the same door handle? The same is true in all public spaces where we're starting to look at hands-free design. There's a lot of innovation and opportunity that we spent time talking about what might be a positive mode. And what it really leads us to believe is that the recommendations for research are pervasive. And we really, we need to take the opportunity to sort of emphasize the power of a research agenda that is national and engaging the research community in this subject area, which is really has, other than you can see in our sponsors, there have been some real commitments to the microbial environment, but it is not a pervasive research agenda across the nation in any way. The Sloan Foundation was one of the major sponsors, EPA, the Environmental Protection Agency, NIH, NASA, and the Gordon and Betty Moore Foundation. This should be a sort of a list of 1,000, a cast of 1,000 who are committed to understanding the microbial environment, because it turns out it's a significant factor in both the positive health and the negative health of the nation. So with that, I think we've done our slides. Is that right? We have. And we'd be more than willing to take questions. We'll pass them back and forth between us, see who is the best person to answer. Thank you. There's always got to be an icebreaker here. Somebody's got to make up a question.
All right, so I'm going to ask you all an icebreaker. So given all of the attention to these devastating tropical storms and hurricanes, we've had Harvey, we have Irma, there's Jose. There's been a lot of attention now to these storms, to the storm surges, and to flooding in people's homes. What does the report say, or does the report speak at all to things that might be relevant to people in those communities who are thinking about how these weather events affect their homes and their health?
So I'll kick off in terms of a response. The committee noted that for years it's been known that water damage in buildings is a risk factor for poor respiratory health. Though repair and maintenance of buildings, particularly in the time of disaster, can be a challenge, and there have been studies of efficacy of measures to repair after Katrina, for example, and then to improve respiratory health, and it makes sense, the committee feels that it makes sense to systematically track which measures to improve the buildings and to improve subsequent health after these disasters are more or less effective. Do any of you have additional comments?
I would say that under the interventions chapter in the report, it mentions that we understand that intense remediation after a flooding event, taking out the walls, replacing walls, going through and changing the room and the buildings in a very thorough way, has been shown in many cases to reduce the health risks, right? But the question then becomes is, what are the cases where it doesn't reduce the health risks? When does a building really need to be remediated? Is the damage minor or major? And if the building is remediated, what kind of tests and assessment can we do in order to determine whether that building has been remediated in a thorough way? And somewhere where, you know, this new science, well, it's not a new science, but the new application of molecular biology and microbial ecology to building sciences comes in is, I think that there's probably some, the report mentions that there are opportunities to better be able to assess whether a building has removed its contamination from moisture, microorganisms growing due to moisture, and has returned back to its natural microbial ecology. And so I think there are some hopeful things in the report about a pathway to better assess building remediation success and the need for remediation.
And let me add in something here as well, unless we have a set of questions that we should be, no, we're okay. Okay, so there are two other issues that come up. One is, of course, the water, the impact of deluge water and the damage to materials that lends itself to mold and other, fungal growth that has respiratory health. There is also the concern about biologicals through compromised plumbing, sewage systems, that all of a sudden you have biologicals that are showing up in the indoor environment that are of serious concern. And something that Hal Levin, if he were here, one of the U.S. specialists in indoor air chemistry would have said is that what people are doing to remediate for mold is often taking chlorine bleach and other kinds of chemicals into the built environment, and those have significant health issues. So we have to be very conscious of the fact that the cure may be more serious than the disease, or at what point, what kind of cures are actually both beneficial to humans and to minimizing risk.
I would like to say that we're talking about not the immediate response, and if there's anybody listening who's in the midst of the current disaster, there are directives about when not to go back into your own home and when to go back into your own home from government agencies, and their EPA and American Thoracic Society recommendations. We're talking about the period that comes after the immediate disaster. And we also mentioned in our report that some of the most vulnerable people have fewer resources and less control over their own building or building maintenance. And as we give our recommendations, we call attention to the fact that these vulnerable populations need special attention.
I've been a practicing allergist for about 40 years and had a lot of experience with from sick buildings to, of course, environmental effects on allergies. And I'd like to commend the panel on the comprehensiveness of the items that are related to human health. One thing that was quite prominent maybe 20, 30 years ago, people were aware that they can't open windows in their schools or office buildings, and they're completely at the mercy of the humidity, the amount of ventilation, which affects the amount that viruses are transmitted from one to another, and the humidity. And we had one school in particular that had some building flaws that didn't allow adequate ventilation, and some of the fumes from their boilers to heat the school were being sucked back in the air intakes. And so there are a variety of building problems they can see. One thing that I think that hasn't been studied as much, at least I haven't seen results, is the effects on the microbiome. Microbiome is affecting probably all aspects of medicine, from intestinal disorders to respiratory problems and skin disorders. I might ask the panel if there is much information available on how the environment does affect the microbiome of humans, either skin or intestinal, respiratory.
Just to understand your question is how the indoor environmental microbiome affects the human microbiome, and that's an important research question. We don't know, though we do know that there are associations, for example, between the microbiome of animals indoors and the microbiome of humans, but research into this area is fairly new. Is that a response? Or
Yes, there's certainly an open field for research. I'm sure it's going to be very important.
right, and as I said in the health part of this presentation, we're not certain that all of the effects of the environmental microbiome relate to colonization of humans as opposed to interaction with human cell surfaces and the airways, in the mouth, on the skin, et cetera.
Yeah, we did spend quite a bit of time trying to uncover what would be good microbiomes, and you know, a lot of the, and maybe some other panelists would like to talk about the probiotic debate, but it does seem as if that there's incredible innovation in medicine that might occur if we begin to understand the microbiota of our gut, or our lungs, or our saliva, or our skin, and being able to begin to heal it if it's flawed. And the physical environment may play a role in that. And as Dan was saying, it looks as if animals, not just indoors, but farm animals and time with farm animals, might be actually a probiotic or a positive force. Now, you mentioned something else that we spent quite a bit of time talking about, and is a particular issue of mine, which is sealed buildings. And I see a growing commitment to sealing buildings. A lot of elementary schools, which once had windows that open, now have new windows that don't open. Hotels where you can't open a window at night, and we are extremely reliant in those buildings on a well-maintained and well-designed and engineered mechanical system to guarantee the airstream, both the amount of dilution we can get from the ventilation air, but also just to make sure that the airstream is not a conveyor of things. And I find that vulnerability, or that dependency, not healthy. But the research is not. There is research going on in Europe, because they're still debating whether to start sealing buildings. So Europe and Scandinavia are still in the mode of keeping every building has windows that open almost. So there's still some research going on in Europe, but very little research in the U.S. about the necessity of us seriously looking at operable windows and natural ventilation as something that's a companion to mechanical systems, not to throw away the mechanical, but as a companion. And the only glimmer on the horizon is that resiliency in the face of these huge storms and fires and earthquakes is beginning to look at, what do we do when the power goes out? And if the power is gone for weeks. So I think there's a little bit of pressure for us as a research community to start to capture the importance of ventilation quality.
Hi. The report talks about a fundamental research infrastructure for understanding built environment microorganisms. Like, I recall some recommendations about a data commons. Why is that important to move this field forward?
I can maybe say a few words about that. There's a number of issues related to why some kind of data commons or infrastructure is needed. I think to begin with, at one starting point is the complexity of the type of data that's being collected now is sort of at an unprecedented level, and particularly with the advent of genomics technology for sampling genomes and transcripts and other molecular measurements of the microbial communities. Generates a lot of data and a lot of very complex data that's difficult to sort of manage and organize, and it creates reproducibility issues and access issues. So it's one of the things that's sort of we have an opportunity to kind of build on existing efforts. The NCBI, National Center for Biotechnology Information, has certainly been a leader in this area of maintaining repositories for large amounts of data that are publicly accessible, but this allows you to sort of divvy up some of the research work, so you can have people developing measurement devices and data collection devices that are depositing information, and then support that or have that applied to modelers and computational scientists that can take that data and build predictive models from that data, those data sets. So that's one part of it. There's also been a recognition in the report that there has to be a continued effort in terms of putting out metadata that describes the sampling conditions and the types of information that are being collected in formats that can be accessible and linked to this raw data that's being collected, so that you can actually do proper cross-study comparisons. And so ideally, part of the issue here is that there are so many parameters to be measured that there's a need to actually start to build across multiple studies, so that you can actually draw conclusions or get more statistical inference power by looking across more than one study to support these efforts. So I don't know if that sort of addresses that issue.
Got a few different questions from the web. I'll start with this one then, which really is what would arise kind of as the end result of people taking on this research agenda and sort of achieving this recommendation. So the questioner is sort of saying people are already designing buildings that have things like humidity controls, but I think it would be really what's the future vision should this research agenda be achieved? What would that enable in the future?
So there is actually a number of discussions that occurred where we talked about what would be the vision for the future of a sort of a healthy microbial indoor environment. And it immediately crosses boundaries with a healthy chemical indoor environment and a healthy physiologically indoor environment. And a number of issues that I think are going to be an interesting question is the amount of time we spend outdoors versus the amount of time we spend indoors, and the power of families and children and people getting outdoors. So I think one of the issues is going to be the need for physical activity and mobility across space. Managing water, there are a lot of issues related to how we're going to design buildings to avoid the constant wetting and destruction that occurs, whether it's buildings that have porous ground floors or ones that have materials that are easy to maintain. So there are a number of things that relative to fungi that I think we're beginning to understand. Much harder to know what the vision of the future is relative to viral and bacterial. I don't know if any of you want to talk a little bit about that. I think we're at the early stages of microbial research, so coming up with the vision of what the ultimate house or office or school is going to look like might be premature.
I would add that if multiple people continuously were getting sick from drinking water, there would be a strong response. We seem to take the fact that we get many respiratory infections each year, that children and adults suffer continuously from allergies, and the fact that many of these exposures occur indoors, we seem to take that as a matter of course versus something that can be better controlled. Because most of our microbial exposures occur in buildings, I think there are many, many examples of where we could improve human health by improving the design of buildings.
Let me get another one that's come in from the web. So the question is about the report summarizing key findings that are relevant for different groups of kind of practitioners or stakeholders. So really the question is, does the report summarize key findings relevant for these different groups? And could you speak a little bit about what some of those key findings might be that are most relevant to some of the different groups of practitioners who will have to be involved in advancing this research agenda? Are there ways that those groups can contribute?
Absolutely. I think the issue is not that we found findings for each of the group, but we found research needs for each of the group. We found where there is linkages. One of the areas that I think we emphasize quite consistently, or at least shows up in a number of the chapters, is the importance of the maintenance and management of buildings and the engagement of occupants in that process. So, facility management. The best intentions can be quickly compromised if the resources aren't there to maintain the building. And the same is true for the way in which you can design a building to provide a healthy indoor environment, and then just the way in which occupants use it over time, whether they're bringing in, whether pests are emerging because of cleanliness processes and pests bring with them their own microbial community, or whether the occupants are using, chemicals and other things to try to get rid of pests, and we start to see compromised environments. So I think probably the biggest chunk of time was spent on that end, or that from the moment the building is handed off, and how it's managed from there. Going backwards in time, Andy Persley was another member of the committee and is at NIST, a leader in the standards development, and certainly in the Airstream. There's huge work that's been going on at ASHRAE and other organizations to try to improve the commitment of building engineers to certainly a healthy indoor air environment, and with that is some of the microbial issues, but there are still a lot of unknown factors. So I think the answer is yes. We do try to touch on what decisions architects make, what decisions mechanical engineers make, what decisions site landscape architects might make, and then what decisions the building owners, managers, and occupants make.
Hi. One of the questions I have is deals a lot with the variation that exists, geographical, cultural, individual variation, and soon temporal variation with climate change. So kind of how metadata will be key in terms of, linking the conditions of particular study sites, but how do we build in flexibility into the research system to kind of adjust for this changing environment?
Well, I'm going to go from metadata to a very specific example of honing in on a particular community, which was the community in New Orleans after Katrina, where there was a community-based research done with the support of the NIH and I believe the CDC to target research on what the best interventions might be to reduce mold exposures and improve the health of children who are asthmatic in Katrina. And that's been written up as an example of how to how to use what's known generally, but focus in on specific community conditions and housing conditions in the midst of an ever-changing response to a disaster. So I think that this is possible, and researchers are aware of the need to be specific as well as general.
I like that question about flexibility, and I think that this has always been a problem in studying buildings. Every building is different, and even if they were the same, they're in different climates and different geographic regions, and that, we know, influences the microbial ecology within those buildings. There are two ways that people have sort of tried to take on this, and the first one is that at some level, everything needs to be studied. So you have to study different geographical regions, you have to study different income levels, and you have to recognize the important differences within those. And so there are broader methods that use more statistical approaches, and I think that databases and use of metadata can improve those studies significantly. But I also want to stress the importance of fundamental science and fundamental research in this area. You know, a large part of the way that microbial communities, for example, assemble in buildings and how people are exposed to those microbial communities deals with the physics of a biological particle or any particle in the air and how it moves from surface to air, et cetera, how it's filtered, how it decays, all of that. And the laws of physics are the same in every geographic region. They're the same regardless of economic status. And so there is still fundamental research that can be applied broadly, and they're still very important to continue with that type of research.
Okay, we've got another question from the web here, which is on communication. So you talked a bit about sort of translating from research to practice, and the role of what I think will be communication in that process. And is there anything that you could expand on communicating to different communities that will eventually need to be informed about the research agenda and its results?
That's a tough one, because this is not the research findings report. This is the research needs report, even though we did as much uncovering as we could to prepare for research gaps in the knowledge. You know, I think there are a couple of issues that are going to be key. I was delighted by the commitment of the medical and health community to understanding how buildings might play a role, and I don't think you see very often research projects that cut across disciplines that way. And part of it is just a paucity of research funding. In some respects, you know, if we could dream, we would make sure that the National Science Foundation or the National Institute of Health set up a long-term commitment, the way they do for many things, to understanding the impact of indoor environments on human health. And that long-term commitment would be underpinned by addressing both fundamental science and to some extent applied science because it's in the built environment, so it's quite applied, through collaborative teams, where you would almost always assume that the teams would have expertise in building science, expertise in health outcomes and social science, as well as expertise in medicine and research related to these fields. So I think the biggest communication that this report needs to have is to the research funding community, to actually help put together those guidebooks that would in fact address. I think your point is well taken. Many of these things become quite geographically and climatically and economically more critical in certain areas, but I think we're at a point now where we need to have that solid research. There certainly are pieces out there that could be communicated, but I don't know.
I think our our chair, Joan Bennett, would really have appreciated that question. The other side of communication is making sure that we understand how what we've just said is understood by different stakeholders and communities. And Joan mentioned the importance of social scientists or others who also, I would say others in the community who can help communicate their needs and their perception of what we're saying about the basic research agenda, or the research agenda in what we would call basic science, as well as the immediate public policy research agenda that we've suggested. And it isn't always easy to clearly communicate why we're interested in stage of life or route of encountering the microbes, or why genetics might modify response. But if we can't communicate it, then it's hard also to convince people that it's important, and we all believe that what we've done and what we've said is important.
One other thing just to add is I think that the report did highlight a number of citizen science projects, which is more of on the area of engagement, more so than communication, but it highlights at least the potential to engage young students in particular, people at an early age, to actually actively engage in collecting samples in their own homes and start to be engaged with some of the questions about how these microbial communities may impact them in a personal way.
Yes, along the lines of engaging young people and hopefully engineers and professionals of all types is maybe a guided challenge on, air sniffing for a building, the breadth of a building, what's in there, and what proportions. Then I also have a question about the neurocognitive effects that was mentioned, and I think I'm aware of some things to deal with molds and so forth, but are there any anecdotes that drive some of the pressing nature of getting this research funded, and any interesting anecdotes? That speaks well when you talk about engagement. The mysteries, what are the mysteries, especially on neurocognitive? Thank you.
So I'll try answering that a little bit. Jean Cox Gensler would be able to answer it in more detail. Most of you have heard the phrase sick building syndrome, and we know that in many buildings, some buildings are considered problem buildings where there's a higher rate of headaches, or people have problems concentrating, or people get more depressed, or children in schools can't perform as well at neurocognitive tests. And from Barcelona, there have been a few published studies suggesting that air pollution, when air pollution is higher, particle pollution is higher near some Barcelona schools, and that adjusts for socioeconomic status, that the ability to perform neurocognitive tests may be lower. But we don't really understand the contribution of indoor microbes per se to neurologic responses or neurocognitive responses. So we listed that as an area of active research.
Yeah, I think one other sort of area of, not necessarily well, in some respects a mystery, but certainly a vision that there are researchers beginning to look at, which is, again, as I mentioned before, the importance of spending time outdoors, but not just outdoors anywhere, but outdoors in vibrant, biodiverse, three-dimensional landscapes. And so there's early childhood development studies that are looking at kids who play on hardtop playgrounds for limited periods of time, and kids who spend, a half of their day out on three-dimensional playgrounds with rocks and trees and landscape of different dimensions, and the impact it has on their cognitive development and their physical coordination, and a whole series of early childhood development indices that are very positive. I mean, it's becoming very clear, again, Scandinavia and places where they actually have a sustained funding stream for understanding what the built environment might mean to physiological and cognitive development. They're beginning to push more and more for the outdoor classroom, and not being a flat, boring outdoor classroom, but something that's incredibly rich and dynamic.
If I think of some compelling narratives, I can put it into two kind of broad categories, but the first one is just the emergence of infectious disease. So we think about new emergence of strains of viruses. We think about different routes that those viruses can cause infection, and think about antibiotic resistance in microorganisms. And so as those strains continue to emerge and we have threats, our exposures, again, are in buildings. And so understanding what can be done in the building to reduce, for example, hospital-associated infections continues to be important and a compelling area of research. The second one is this idea of a healthy microbiome. And the more that we understand from human microbiome research about how our human microbiome is important in the development or lack of development in many different chronic diseases. Again, much of our exposure to microbes is in buildings, and so understanding that key part of the equation, which is, okay, so if we understand what are the microbes and the human that are important to preventing these diseases, understanding how they acquire those and the role of buildings in that acquisition is important.
All right, I'm just going to remind folks listening on the web that if you have questions to ask, you can email them to the builtmicrobiome@nas.edu, the website that's on the screen. And we have another one that I think goes back a little bit to what you were previously talking about in terms of the research infrastructure and the kind of needs in that field. So the real question is, how do we know what the real microbiome is with all the variability that's reported? And that's really reflecting things like variability in results as a result of different DNA extraction protocols, variability in the results from different ways that the samples are prepared, or the bioinformatics processes. So for those in the room maybe who are less familiar with some of that, maybe you could speak a little bit to some of those sources of variability and what that question is really referring to, and then how would one think about making progress and maybe tackling that issue, kind of a little more detail around what you're talking about cross-study comparison there.
So there's many sources of variability in the measurements. There's sort of the biases from DNA extraction methods. So depending on the particular type of organism that DNA extraction is being operated on, may be more or less amenable to particular extraction methods. So it's important to actually articulate and clarify specifically the types of sample extraction tools, kits that are being used, so that can be at least appropriately documented. So there's really actually sources of variation that can potentially go, through each of the different stages. So that has to be documented as you go to sequencing. If you're doing a sequencing type of measurement, there may be some biases in terms of the nucleotide composition that suggests which fragments are more relevant or privy to being sequenced and represented. And then finally, on the database side, in terms of identifying the contents of the organisms by matching the fragments to reference sequences, that the reference sequences themselves are continuing to evolve over time as better representation is being collected and stored. And so those types of assessments about the particular strain or particular taxonomic unit that are being identified may evolve somewhat over time. So it's important to actually be able to store as much of the raw data as possible, number one, so that you can go back and re-evaluate or re-extract information based on different methods and sort of to verify and validate that those results are reproducible in terms of the type of community composition that you're asserting are present. So that's one sort of key aspect of it. And then, so I would say that I'm kind of losing my train of thought in terms of the direction of where the question was going. But so that kind of shows some of the technical variation. There's also the biological variation, just because of the amount of low biomass and sort of the diversity of organisms present. It could be easy, just from a sampling perspective, to capture different. And you may need to do repeated sampling to actually get a statistical average of what's actually present. So the ways to kind of address that, I think, are, or some of the ways to address that, I think there's more effort going into sort of building reference material, and some of this effort is being sort of led at NIST in terms of building compositions of microbial community organisms that could be used for benchmarking, so that you could sort of test different techniques and methods and assure that the same results are being generated, or at least to provide some sort of standard variational measurements that you would expect to see for a particular set of extraction and analysis tools that are applied. So I think that sort of effort to build better benchmarking and reference materials is one important part, along with sort of this idea of having the raw data stored that can be reanalyzed from scratch from multiple tools and toolkits to verify recreation of the same results. And
I was going to say, if I think about a case where it could be compared, so for example, thinking about chemical standards in drinking water. So there are laws, and there are drinking water standards for specific chemicals. And because of that, there are specific methods for determining those chemicals, concentrations. It's different than what the study of the indoor microbiome has been thus far. It's mostly been done by researchers at university and other private labs, and they all use their own method, and they don't want to use a standard method because they're interested in innovating, and they probably think, we all think that our method is the best. But there's no doubt that if this research progresses into some types of standards, for example, a standard mold test, then standardization is going to occur and will need to occur, at least specifically for those types of tests.
Could you address in a little bit more detail what the state of research is in understanding what constitutes a good indoor microbiome, and steps that could be taken to maintain or improve good microbes within the indoor environment?
I'll start with broad brushes. So I know that many don't like the term healthy microbiome because we don't particularly know what a healthy microbiome is. It's a good concept, though, but less well defined. There's some understanding, and there are many different names for the hypotheses, the hygiene hypothesis, the old friends hypothesis. There's some understanding that humans may be missing, at certain points in their development, microorganisms that we used to be exposed to. Right? These are microorganisms that we get from animals. These are microorganisms that we get from outdoors in the environment. These may be microorganisms that we get from our mothers. These may be low-level helminth and worm infections. And so to the point that we can encourage those microorganisms, maybe not worm infections, but certainly exposure to outdoor air in the outdoor environment, And in some cases, but it's tricky, exposure to animals, for example, pets in homes, we may be able to generate a microbiome that's more healthy, but it's different for every person. For example, if you have allergies to dogs, probably getting a dog for a healthy microbiome is not a good idea.
Sorry. Go ahead. So one of my microbiology colleagues has said to me, there's no specific good microbe or bad microbe. It's just the amount you're exposed to, what company they keep, what your genetics are, et cetera. But I think we do know about specific microbes that that can be infectious. We know a fair amount about the transmission of influenza, tuberculosis, C. difficile, but we really don't know that much about what combination of microbes would generally be protective. And the study of good microbial situations has led to some extent to the identification of some microbial components which might be common across a number of different taxa of microbes that could, if encountered at the right time of life by the right hosts, and it could be a large proportion of the community, would start responses in the immune system which might protect against allergy and allergic airway disease. Now, as we do more research about what good microbial situations are, we'll learn more about how to apply this research either to the creation of buildings which are generally healthy for most people, or ventilation systems that are generally healthy for most people. And we'll also probably find some information about components of microbes which could be packaged in one way or the other and be protective for very specific groups. I tend to back away from identifying particular taxa that are good, but I think we're in the middle of a very exciting time in terms of trying to find communities or situations which could be protective for many people, and then also components of bacteria or fungi that might be protective for some people but not for others.
Before you go to another question, I think there's one other issue that a study that we discussed in one of our meetings was one that looked at child care facilities and the number of colds and flus that kids get from each other in child care, and of course this whole, and which they bring home, and then you have them as well. And it turns out that in one study they have found that over a longer period of time, these kids are actually much more resistant to colds and flus. So as adults, you have fewer colds and flus. So maybe it's a microbial vaccine that you get in childcare that begins to make you healthier as an adult. So these studies really do need to be longitudinal, especially in the area of good microbes, to understand what might actually be a long-term benefit.
Maybe I'll first find out if anyone else in the room wants to have a question. Otherwise, I'm going to ask maybe the last question for the panel, and we'll close it. Okay, so as the final question then, for you four that are sitting up there on the panel, if there was a sort of one or maybe two kind of take-home messages or points that you wanted people to kind of remember from this report, would you be able to comment on kind of what would be your take-home message from the report to the audience?
Okay, I'll start. There were only a few of us who were building scientists in the committee, although everyone is touching on building research in the committee. And it is such a vast, first place, it's such an unknown for most architects and building engineers to really understand the microbial environment and its impact on health. So there's just, and trying to amass the literature and starting to read the literature on microbes in the built environment. The one thing that I think helped clarify it for me, which was sort of an aha moment, was to actually try to differentiate between things, the microbial that are waterborne, microbials that are airborne, which of course they can cross the boundaries between the two, and microbials that are exchanged from human hand to human hand through surfaces or from other living beings to surfaces and back to the human. And so that differentiation of what is the microbial water, what is the microbial air, and what is the microbial surface environment was a really good aha moment for me to understand, at least to begin to have buckets for the research as I was looking.
I think that this committee really was a treat in terms of the interaction between architects like Vivian and microbiologists and building specialists and myself as a health expert. And I think I would have two messages. One would be in the applied area and one in the basic mechanisms area. We need to improve our understanding of what we're doing to the microbes when we change building and have better communication with those who manage buildings. And then understand the relationship between what we've done to the buildings and the microbes and what microbes are in and on people. And then the second message is that we need to learn more about the mechanisms through which environmental microbes affect human microbes and therefore human health.
I think that what I take away from this report in this subject area in general is that with very few exceptions, we all live in buildings, and we spend the majority of our time in buildings. So this is a topic, unlike many other topics, that touches all of us. And so given that many of our exposures occur in buildings and that they occur to all of us, and that we know that there are defined and undefined problems with microbial exposure in buildings, that this could be one significant way to improve human health.
The one thing I guess I would add from my data science perspective, I think, is that I guess given the kind of complexity and the number of parameters and variability of what we're trying to measure here in terms of the communities and microbial communities as well as the built environment, there is a real opportunity to sort of take advantage of the growing data science field that supports these opportunities to share data, to share analysis tools, and that's going to be something that can be potentially really useful for this field to sort of help manage the complexity of the data that's being generated.
Thanks, Diane. Thank you all for coming, and those on that are on a webinar, thank you for joining this meeting. And I know that you will be able to read the report and probably send more questions back through some other vehicle as time goes on. Thank you.